Hey, Hammy here, back with video number 2 from chapter 8, in which we will look at the first step of cellular respiration, which is glycolysis that is happening outside the mitochondria. Okay, let's talk about step 1. Step 1 is glycolysis. Okay, glycolysis, if you look at the root of the word, glyco means sugar, lysis means to split. So we are sugar splitting. We are splitting the sugar in half. It's occurring outside of the mitochondria. It's in the cytoplasm. And it's a series of 10 reactions, each with its own enzyme. If you want me to break out that old biochemistry book from college, I can show you each of the steps and all the enzymes. We will not get that in depth, however. It's important to note that there's an energy investment step and an energy harvesting step. In order to get this process started, you got to put in 2 ATP, okay, in order to activate the glucose to get it started. Think of it as like my brother-in-law always tells me, in order to make money, you got to spend money. Okay, you got to invest a little bit first, then you'll get some return on that investment. Same thing here with our cells. Okay, they're going to invest 2 ATP to get the process started, which is going to split the glucose into 2 G3P molecules. And then it can go through some energy harvesting steps where oxidation of G3P, we're going to remove electrons and hydrogen ions, which are going to be picked up by NAD+. And you're actually going to make 4 ATP through substrate-level phosphorylation. This is not chemiosmotic phosphorylation. This is substrate. So in other words, there's no chemical gradient with hydrogen ions that go through an ATP synthase, the spinny thing, and cause ADP to phosphorylate into ATP. This is where you have an enzyme that's going to take ADP and a phosphate and some high energy and put that phosphorus on it, making ATP. So if you put in 2 and you get out 4, that's a net gain of 2. Okay, so you're coming out to the good, and your end product, okay, the glucose is going to be split into 2 pyruvate molecules. C6 molecule is going to be split into 2 3-carbon pyruvates or pyruvic acid. Okay, you'll see that name, same name for that same molecule. Okay, this slide is just kind of showing the overall inputs and outputs from glycolysis, which was step 1. In other words, you start with 1 6-carbon glucose. It's going to be broken into 2 3-carbon pyruvates. Okay, as it's broken apart, the electrons are going to be picked up and used to reduce. Okay, glucose is going to be oxidized into pyruvate. NADH is going to be reduced into NADH. In other words, we added electrons. Okay, so again, redox reactions. Okay, here's that keeps haunting us, right? We're going to invest 2 ATP, but we're going to end up with 4 ATP, which is a 2 ATP net gain. So this first step of just cracking glucose apart, we're going to get some high energy electrons up here, which are going to be taken to the ETC eventually to be made into more ATP, and we're getting 2 ATP directly, a net gain of 2. Okay, if we kind of break down these steps, again, we're not going through all 10 of them, but if we kind of break it down as simply as we can, okay, here's the first stage of glycolysis, the energy investment step. Okay, so negative 2 ATP, we're going to put here and here, we're going to put in 2 ATP. Here's our 1, 2, 3, 4, 5, 6 carbon glucose. Okay, it's going to be broken down. Okay, we're going to add the phosphate group. So adenosine diphosphate and triphosphate. So we're going to add a phosphate group to each end. Okay, and then it's going to be split. Again, there's enzymes that do all this. It's going to be split into 2 G3Ps. Say, that looks familiar from last chapter when we were talking about photosynthesis. We used 2 G3Ps and put them together to make glucose. And now we're kind of going backwards here, okay? And if you look, G3P, glyceraldehyde 3-phosphate. Remember that 3 tells us where on which carbon that functional group is attached. So here's carbon 1, carbon 2, carbon 3. That phosphate is attached to carbon 3. Again, because of the way it's just showing it here, this is actually carbon 1, carbon 2, carbon 3. There's the phosphate attached to carbon 3. And that's kind of where we get that name from. Once we get the two G3Ps right here, notice it was split twice, so we're following this. It's, both sides are the same. They're just showing both pathways. So you could just show one pathway and then times 2, right? Because you have two G3Ps. Okay, the second part of glycolysis, part B, is the energy harvesting steps. So now that we've invested energy in to break it apart, to activate it, turn it into G3P, okay, we're going to start getting energy out of it. Okay, the first step here, we're adding another inorganic phosphate group, and we're turning G3P into BPG. Okay, now look up at BPG up here. It's 1,3 biphosphoglycerate. Okay, 1,3 again tells us where the functional group. So here's carbon 1, 2, 3, and here is carbon 1, 2, 3. So notice 1 and 3 both have the phosphate groups attached to them. Now when that happens, off comes electrons, which are picked up by the coenzyme NAD+ and turned into NADH. Okay, so we've harvested a little bit of energy, not ATP, but we've harnessed a little bit of energy in the form of high energy electrons. And again, these are going to be taken to the electron transport chain to be made into ATP, which the cell can use. Continuing on with the energy harvesting steps, the BPGs are now changed into 3PGs, a 3-phosphoglycerate. Okay, again, third, here's carbon 1, carbon 2, carbon 3. That phosphate is off the third carbon, hence the naming, okay, of the molecule. And notice when that happens, when we go from BPG to 3PG, this high energy bond is going to be used to phosphorylate, substrate level phosphorylation of BPG, okay, into ATP, okay, and again over here as well. Okay, so we're already even. Okay, so after a couple steps here, we've invested 2 ATP into the process, right? And now we've gotten those 2 back. So we're back to the good. We're back to even. Okay, continuing on here, the 3PGs, okay, are then you're gonna remove a water. Okay, you're gonna oxidize. You're gonna add, remove some water. You're gonna add some electrons and change them to PEP. Okay, gonna change them to PEP. And if we look at the next step, way down here, we're going to go from PEP down here to our final 2 pyruvates. And you see when we do that, there's our substrate level phosphorylation or substrate level ATP synthesis. Here's 2 more ATP. So we have 1, 2, 3, 4. We've harvested 4 ATP, 2, 4. And we put in 2. We get out 4. That's a net gain of 2 ATP. Okay, so this first step of photosynthesis, glycolysis, which happens out in the cytoplasm, just about every creature on Earth, every living thing, fungi, plants, animals, bacteria, just about everything we know does glycolysis. We think this is one of the rudimentary first processes in harvesting energy from carbohydrates and living things, is taking glucose and splitting it apart. Okay, you're not getting a ton of energy, but it can happen quickly, and you can get, it doesn't need oxygen, it doesn't need a special organelle. You just got to have the right enzymes to go a few steps, and you're going to get a net gain of 2 ATP. Okay, so as long as you have sugars to break down, you're going to be getting 2 ATP, 2 ATP, 2 ATP, 2 ATP, and just about all living things do this. The last slide here is just kind of showing a substrate-level ATP synthesis or substrate-level phosphorylation. Okay, where you've got an enzyme that's going to, so here's one, for example, when we went from BPG to 3PG, okay, we made an ATP. How does that happen? Well, it takes this phosphate, okay, right here. This is this high energy bond right here, right there. And it removes the phosphorus in that high energy bond. So the enzyme right here would be what? The active site, right? So it has an active site that will bond to BPG and cause a conformational change and help with the moving of this high energy bond from BPG over to ADP. Okay, and when it releases, the products are going to be 3PG and ATP. Okay, so this is done without any chemiosmosis, any pumping of hydrogen ions like we see in the electron transport chain. This is happening directly by an enzyme, okay, at the substrate level, taking the phosphate and the high energy bond off of one molecule, an organic molecule, and putting it onto ADP to make ATP. All right, so that's glycolysis in a nutshell. In the next video, we'll look at what happens if there is no oxygen for this to continue. It does not go into the mitochondria.